Thanks for inviting me, and I'm really happy to be here and get a chance to tell about some of the things that we do in JBEI with cell wall biosynthesis. And I know a lot of you who are also working with cell walls, but I see many faces that I have not seen before, so I'm happy to be able to convey what we do to more of you. So this is just a picture of all the people in JBEI, or at least all the people in JBEI who would come to this retreat. We are probably about 160 people out in JBEI now. I need to acknowledge that JBEI is a consortium with 6 different partners. So Lawrence Berkeley Lab, which is the host institution, and that's where most of us are employed, but there are also people from Sandia and Lawrence Livermore National Labs, and from UC Davis, from UC Berkeley, and Carnegie Institution of Science is a partner, although they don't really have any people at the moment who are there. So we are interested in JBEI in this process of converting plants into fuels. And the method that we are investigating, and this is the mandate we have from the Department of Energy who is funding our project, that is to use an pre-treatment, enzymatic conversion, microbial fermentation into fuels. You can make fuels from biomass in other ways, but this is the process that we are studying, and that's what DOE funded us to do. So this is what we look at. And we are not really looking at how to grow the plants, and we are not looking at other types of conversion. We focus mostly on fuels, but of course this platform is also very suitable for making other kinds of products that can substitute petroleum-derived chemical products. We are divided into 3 scientific divisions: a feedstocks, a deconstruction, a fuel synthesis division. And then we have a technology division, which are, well, developing new technologies and also to some extent function as a service platform for the scientific divisions. So I'm in the feedstocks division, and we are interested in the plants and how can we change the plants ultimately into something that will be better feedstocks. So plant biomass, that consists basically of cell walls. And you can find models of the cell walls like this in papers. There are different kinds of cell walls. These are primary cell walls, type 1, which is what you find in most plants, and then the type 2 walls, which you find in grasses. They have a somewhat different composition. And again, you can find these models, but I need to say that we actually don't really know that it looks like that. It probably doesn't look like that. All these cross-links and interactions between the different polysaccharides, they are not well understood. So there's a lot of imagination in these models, and I think it's important to keep that in mind. Now, the plant biomass that we would want to use for biofuel production doesn't actually look at that at all, because what we are mostly concerned with in biomass to biofuel conversion, that are the secondary walls, which are thickened walls that form in some cells when they stop to expand. So that's what you have, most of the wood and also the stems and grasses, that they contain a lot of secondary cell walls, and they make up most of the biomass. The composition is something like this. So we have maybe a third of cellulose, a third of hemicellulose, which is mostly silane in the secondary walls, and then lignin, maybe a quarter of it is lignin. So this forms some challenges for us when we want to use it to convert it into fuels, because this kind of secondary walls with this composition, they're not very easy to degrade. We need to degrade them into sugars if we want to use fermentation route to make products. So first of all, they're not very easy to degrade because of cross-links and because of the presence of lignin. The sugar composition is not optimal. There are a lot of C5 sugars, pentoses in the hemicellulose, which are not that good for fermentation. There are acetate and ferulic acid esters, which inhibit fermentation. And then we have all this lignin, which makes it difficult to degrade the polysaccharides. And when we have degraded the polysaccharides, well, then you still have the lignin, and it has very little value. So, this is up to a third of the biomass, something that you just have to transport around, and it doesn't really have any value. So those are our main challenges that we keep in mind when we study the biosynthesis of the cell walls. So the cell walls the polysaccharides in the cell walls, they are made in the Golgi apparatus, except for cellulose, which is a lot of it, but which we don't work with. So I'm not going to talk about cellulose. So we are concerned with the things that happen in the Golgi. And this is part of a figure that I've stolen from Marcus. I hope that's okay, Marcus. So the glycosyl transferases are the main enzymes that produce the polysaccharides. So most of the glycosyl transferases, they are type 2 membrane proteins. They have a single transmembrane region and a catalytic domain in the lumen of the Golgi. Then they use nucleotide sugars. Some of the glycosyl transferases are multi-membrane spanning proteins. We have in Arabinose, which we mostly work with in my lab, we have about 470 different glycosyl transferases, maybe more. We can find database here. Most of them, we don't know what these glycosyl transferases are actually doing. We estimate that maybe we need some 150 different glycosyl transferases to make all the different polysaccharides and glycoproteins in the wall. We don't need only glycosyl transferases. We also need methyl transferases, acetyl transferases, and ferulyl transferases to make the polysaccharides. And then we need other things, nucleotide sugar transporters, nucleotide sugar interconverting enzymes, and also hydrolases, which modify the polymers after they're being synthesized. So altogether, it's a lot of enzymes that are and proteins involved in the biosynthesis. Just to give you an example of the complexity of polysaccharides, this is a schematic structure of pectin. There are different domains in pectin, many different sugars and sugar linkages, and we estimate that perhaps we need something like 67 different transferases to make all the different known linkages that we find in pectin. And we really only know 3 of them. We know the enzymes that put this xylose there and that xylose there, and the enzymes that make the backbone. But all the other ones, we haven't really found them yet. So there's a lot of challenge, even though pectin is not very much of the biomass. It is important in biomass, and we have all these enzymes in our databases that we need to find out which make pectin and which make something else. So different approaches that we have followed to try to find out what are the enzymes and how do they work is based on the genes that we already know. So we can take the genes, we can find knockout mutants, which is pretty easy in Arabinose, and we can see is the cell wall changed. And then once we see the cell wall changed, we have an idea what the enzyme is doing. We can try to determine its activity. We have looked at a lot of mutants in that way, and we also have some very interesting glycosyl transferases identified in that way. But many mutants, they actually don't show a clear phenotype because of redundancy or because they only change maybe polysaccharide in very specific cell type or something. So it's not the only approach. Another approach that we followed in JBEI is to clone all the glycosyl transferases and try to see if we could establish robust procedures to determine their activity. And so one way that we have used, and we have all these young women have been involved in doing that because it's a lot of enzymes. So one way we're doing it is that we express them in tobacco, which we like to use a plant system because they're plant enzymes, and we feel that chances that they will be active is better than if we express them somewhere else. And then one assay is kind of the robust assumption-less assay is that we, instead of using oligosaccharide or polysaccharide as acceptor, we can use monosaccharide. If you use the high enough concentration, you can, in some cases, drive the reaction towards formation of disaccharide. Then we form the disaccharide, and we can determine the activity in this way. And we have set this up also to work in a high throughput way, at least a 96-well system. This is an example of how we can use that assay to show the activity of this enzyme, which puts xylose on fucose. And this was identified some years ago by Peter Ulsgaard's group in Copenhagen. So in this case, UDP xylose, radioactively labeled, is reacted with high concentration of fucose when we have this enzyme expressed in this case in tobacco. And yes, when we have fucose as acceptor, we have a high activity. If we use xylose as a negative control instead of fucose, we don't have much activity. And this is a control from tobacco plants that are not expressing the enzyme. So in some cases, the assay works very well, but it doesn't always work. So we haven't been able to show the activity of all the hundreds of different glycosyl transferases with this way. We're still trying to do it, but it wasn't as easy as we could have hoped. So we're also trying a more focused approach to study specific enzymes where we have a better idea of what they do. And we are mostly interested in silane, or we are particularly interested in silane because it is the major non-cellulosic component in the biomass. So silane has different composition depending on what plant and what tissue you're looking at. But overall, it's like that, a backbone of xylose, side chains of different kinds. And the side chains that I'll tell you something about are the acetyl side chains, ferulic acid side chains, Arabinose side chains, and glucuronosyl side chains. So we are working with all these and trying to find out how they are being made. So first about Arabinose, the one strange thing about Arabinose is that it's unlike most of the other sugars in the polysaccharides. It's almost exclusively present in the furanose form, not the pyranose form that you find, for example, for xylose. The substrate for putting Arabinose into polysaccharides is this UDP arabinofuranose, whereas the immediate product from UDP xylose is the pyranose form of UDP arabinose. This has been a conundrum until a few years ago when a Japanese group showed that there's actually an enzyme in plants that will convert the UDP arabinopyranose form to UDP arabinofuranose form. This is the immediate substrate for the incorporation into polysaccharides. So we wanted to look at this group of enzymes, the mutases, which are glycosyltransferases in family 75. They're also known as reversibly glycosylated protein because they have this self-glycosylating activity. So there are 5 genes in Arabidopsis, 4 of them in this group here, and 1 here in this class 2 group. So we try to find out what function do these 5 different genes or proteins have, and Carsten Audengarten in my lab is working with that. So he made constructs to look at the subcellular localization, driving fluorescent fusion protein with native promoters, and basically it shows that the RGP1, 2, and 5, they're pretty much expressed everywhere. They're cytosolic proteins. The RGP3 and 4, they're also cytosolic proteins, and they're expressed only in the seeds, developing seeds. So what about the activities? We expressed the proteins in E. coli and set up an assay. So here we had the 2 different forms of UDP arabinose. And if UDP arabinofuranose, which you cannot get, you cannot buy it, well, you can get it from friends like Tadashi Ishii. So if we take this substrate and incubate with the enzymes, then we can convert it into UDP arabinopyranose. And you can see here a control. Of course, nothing happens. This is the RGP1. So when UDP arabinofuranose was incubated in the presence of RGP1, we had a conversion to UDP arabinopyranose. And it goes the other way too. We can incubate with UDP arabinopyranose and we form a bit of the furanose form. The equilibrium is very much towards the pyranose form, so that's why you don't get an equal proportion. And this showed that RGP1, 2, and 3, they have this activity, whereas 4 and 5, they don't have any activity that we can detect. So they don't seem to have this activity, and we don't know why they're there. They form complexes with the other proteins. So we wanted to find out what role do they have, and well, obviously in making UDP arabinose, but were they essential for incorporation of arabinose into polysaccharides? That's what we wanted to find out. It was already known that a double knockout of RGP1 and 2 is lethal, but so we thought we should try instead to use RNAi. Carsten, he made a construct to downregulate RGP1 and 2 by RNAi, and this shows the expression in different plants that he looked at, and some of them you can see they're very much downregulated in RGP1 and 2, and he has RGP5 as a control here. It was not affected by this construct. And it also shows here the same series of plants that the content of arabinose correlated with the transcript level. Again, this just shows only the arabinose, that the content of arabinose in the cell walls of these plants, not only in silane, but actually mostly in other polysaccharides and pectin, correlates with the amount of RGP1 and 2 transcript. And the phenotype of the plants also. When we have plants that have up to, say, a 50% reduction in arabinose, we don't really see a phenotype. They seem to be growing perfectly normally with half the amount of Arabinose. But when we get plants that are below that, then they really start to get very much affected. And the most downregulated plants we had, well, they couldn't really grow. So we had plants that were smaller and we couldn't measure anything because they died before they ever got off the ground. So this tells us, we think that UDP Arabinose is converted from the pyranose form to the furanose form by this enzyme and not by other enzymes, because it has been a bit of a puzzle that we have this mutase and the activity had been shown in E. coli like we also showed in our work. So it's pretty clear that it has this mutase activity, but it is kind of puzzling that it's out in the cytosol externally associated and loosely associated with Golgi. So it's puzzling because UDP arabinopyranose is formed inside the Golgi by this enzyme. And the glycosyl transferases, well, they are also inside the Golgi. So the glycosyl transferases that uses UDP arabinofuranose, they are inside the Golgi. So this is formed inside the Golgi. This is used inside the Golgi, and yet this enzyme that converts the two forms is outside the Golgi. So that's just puzzling, and some people have thought, okay, can we be sure that there's not another mutase inside the Golgi? But since we can completely eliminate arabinose in the cell wall by downregulating this enzyme, we think that there's no way that there could be other mutases inside the Golgi. So it's kind of a puzzle, and we must conclude that there have to be some transporters that can transport UDP arabinose out and back in. Maybe it's the same transporter, maybe they're different transporters, and we don't know that yet. So it's kind of a puzzling way. That's what I would call unintelligent design. So, oh, no, I don't want to show that one. Oh, yeah, wait a minute. I wanted to say here that we were interested to find out, if we could use this protein as a tag to identify some of the other proteins, maybe the transporters or the arabinosyl transferases. So we have expressed them in plants with tags and immunoprecipitated the proteins. And we do find complexes. But the complexes, they only contain IgP1 and 2 and one of the other isoforms. So they form complexes together, several different RGPs in the same complex. But at least with the way we did the immunoprecipitation, we were not able to identify any other proteins. We need to work more on that with milder solubilization. Then we have been also looking at other parts of the biosynthesis. So how does ferulic acid get onto the arabinose? One thing is we need to have the UDP arabinofuranose, but we also need to get ferulic acid attached to arabinose. Again, this takes place inside the Golgi, but how is it going to happen? What are the ferulyl transferases involved? And already some years ago, Mitchell and Dupree, they pointed out that there was a group of acyl-CoA-dependent acyl transferases, which were good candidates for ferulyl transferases. So that was based on expression studies in grasses that they had done. And they identified this family, and here I show all the Arabidopsis isoforms or members of this family, and then the clade of grass unique members of the family. There are many other grass members, but there's this special clade which has very many grass members. There's also one member of Arabidopsis in this grass clade. So this was pointed out that maybe those are the ferulyl transferases. So we have looked at that in rice, and also these closest relatives in Arabidopsis. And this is work together with Pam Ronald and Laura Bartley, a postdoc in her lab who's now in Oklahoma. So Pam is in Davis, and she's doing the rice genetics work in JBEI. And one of those rice mutants actually showed a very much decreased content of ferulic acid. And that's interesting. It tells us that it's the right prediction that these enzymes are really involved in ferulation, but it also has a direct implication for the biofuels. Ferulic acid in the cell wall of grasses cross-links and makes the grass cell walls very difficult to digest. So we could imagine that if we had less ferulic acid, it would be easier to digest the walls. And yes, if we take walls from these mutants that have less ferulic acid, they are easier to digest here with cellulase. So that worked well. Another thing is that ferulic acid is inhibitory to yeast. So the released ferulic acid will inhibit yeast fermentation. So we happy to have plants with less ferulic acid, especially because these plants, as far as we can see, they grow happily. In these greenhouse grow plants, we cannot see any difference in biomass accumulation or vigor of the plants. But what about the Arabidopsis gene? So there's several Arabidopsis genes that show similarity to these rice ones. Now, Arabidopsis is not known to have ferulic acid and the cell wall polysaccharide, but we were not sure if perhaps there was some anyway, maybe in the pectins. So we have looked at knockouts in all these different Arabidopsis genes. And most of them we don't see anything, but we did find some that have less ferulic acid in alcohol-insoluble residue isolated from the plants. So this shows one of these genes. We have 3 different knockout lines. There's no transcript. And when we measure ferulic acid, it's much less. There's no real change in coumaric acid, which is another. So where is this ferulic acid in the cell wall of Arabidopsis? Is it really the cell wall or is it something else? And we don't think that it's in the cell wall. When we try to fractionate this preparation from Arabidopsis, we cannot detect any ferulic acid in the cell walls, polysaccharides. We can detect ferulic acid in suberin, cutin part of the preparation. So cells of plants have lipid compounds, suberin and cutin, both on the cuticle and also in the endodermis of the cells. And these compounds in cutin and suberin, they are very similar. Here's a kind of overview of the kind of constituents they have. So different carboxylic acids, hydroxy fatty acids, alcohols, and also phenolics. There is ferulic acid in both cutin and in suberin. Up to 10% in suberin is ferulic acid. So we thought maybe that's where the ferulic acid is missing in our mutant. And Carsten, he fractionated his plant material doing digestion with pectinase and CDTA extraction to extract pectins, see if there was any ferulic acid there. We didn't find any ferulic acid there. And then he looked at the lipid fraction using a method with boron trifluoride to depolymerize the polyesters and extract the compounds into ethyl acetate, hydrolyze them, and run them on the HPLC. So they were just following the published methods for characterization of the suberin and cutin. And the bottom line here is that this is from a heterozygote plant, so this is like a wild-type control. We find coumaric acid and ferulic acid in this extract, but in the homozygote mutants, we find almost no ferulic acid. So we conclude that from this that ferulyl transferase is probably involved in making suberin in Arabidopsis. And to further prove that, we also express the enzyme in E. coli, but first we had to make the substrates, because again, these are substrates that we cannot purchase. We need ferulyl-CoA, coumaryl-CoA, caffeoyl CoA, synapyl-CoA. So to make those substrates, we took 4-hydroxy hydroxycinnamyl-CoA ligase from tobacco and from Arabidopsis and expressed in E. coli. And with these enzymes, we can produce all these substrates that we could not buy. So we have them. The tobacco enzyme will make ferulyl, caffeoyl, and coumaryl-CoA, but it will not make synapyl-CoA. But there is an enzyme in Arabidopsis which is known to make, or was known to make synapyl-CoA, and it also does that in our hands. So we could make these substrates, and now that we had the substrates, we could express the putative ferulyl transferase from Arabidopsis in E. coli, incubate the different substrates with omega hydroxy fatty acid, and yes, we actually can in vitro transfer all these different hydroxycinnamates to the hydroxy fatty acid. So in vitro, we can synthesize these different products. Actually, in vivo, in the mutant, we only see the ferulic acid is decreased, so we don't see a decrease in coumaric acid. So we don't know why there's a difference between in vivo and in vitro here, but I think it pretty much proves that this is what the enzyme is making in Arabidopsis. So to go back to this one, both this Arabidopsis enzyme, but also the rice enzymes and all the other members of that family, they are predicted to be cytoplasmic proteins, and they also look like cytoplasmic proteins in fluorescent labeled experiments. So now we have ferulyl transferases that are out here in the cytoplasm, and yet the ferulylated polysaccharide is made inside the Golgi. So we have another conundrum here. How does a ferulyl transferase transfer ferulic acid from ferulyl-CoA into a polysaccharide which is inside the Golgi? That's not really possible, right? So there must be an intermediate, and we don't know what that intermediate is. We had thought that perhaps the intermediate would be UDP-Arabinose so that it could transfer ferulic acid directly to UDP-Arabinose, and then maybe it's the whole UDP-Arabinose with ferulic acid that's being transported into the Golgi. But we have not been able to demonstrate the activity of transfer from ferulyl-CoA to UDP-Arabinose with any of the ferulyl transferase candidates that we have overexpressed. So we still don't know what the intermediate is. It might happen in another way that we haven't figured out yet. So another thing that we are working with is acetate and acetylation. Again, we have present in different polysaccharides, in many different polysaccharides, but especially in silane we are interested in. And there can be quite a lot, especially in wood from hardwood silanes. They have a lot of acetyl esters. And why are we interested in that? We are interested in that because the acetyl esters, they will not surprisingly inhibit deconstruction, so enzymatic degradation of silane with a lot of acetate will be inhibited by the acetyl ester groups. But more importantly, the acetic acid inhibits yeast. So you don't get as much biofuel as you would want if you have acetylated polysaccharides. So that's a reason for having less. So we wanted to find the acetyl transferases, or the machinery putting acetyl groups into polysaccharides. And we had some candidates from a study that was done or published in 2001 from Cryptococcus neoformans. So Cryptococcus neoformans has a coat of polysaccharide, and the mannans, the mannose residues in this glucuronoxylum mannan, it has acetylation. And there was a protein, or rather a gene from Cryptococcus that was known to be involved in this. How it does it, that's still not known, but there was this Cas1p protein somehow involved in acetylation. So Arabidopsis has 4 candidates, 4 homologs of the Cas1p protein. There's also homolog in mammalian cells. So we thought maybe they are involved, and we got hold of this knockout Arabidopsis mutants in all the 4 different genes, and just to show you that there's no transcript for these. And then we looked at the cell walls. And with these mutants that we call RWA mutants, reduced wall acetylation, in gene 1, 3, and 4, we didn't really see any significant difference. But with RWA2, we had a 20% reduction in cell wall acetate. It's not a lot, but we have seen it many, many times. We have several different alleles. It just shows how we can cross 2 different alleles and get still 20% reduction. We know that that the mutation is really causing the decrease in acetate. Now there could be less acetate, not because it's directly involved in acetylation. It could be a change in the polysaccharide composition that indirectly caused the change in the acetate. So we don't look at sugar composition, and we don't find any difference in sugar composition in the plants. So where is the acetate sitting? Again, many different polysaccharides can be acetylated. So we had 20% less acetate. Which polysaccharide had less acetate? We wanted to find that out. And one way to try to find that out would be to use antibodies. So we have an antibody from Paul Knox in Leeds, LM23, which recognizes cyclogalactronin and silane when it's not acetylated. Now, the problem that it has this dual specificity, but we have another antibody that recognizes silane, LM10. And here we looked at the epidermis in stems, and there was no silane, so we could focus on cyclogalactronin. And what you see here in the wild type is that we have very little labeling with the antibody, but in the mutant, we actually have a significant amount of labeling in the epidermis. Now, that could be because there's less cyclogalactronin there than there, perhaps, but we can treat the sections with alkaline, and the alkaline will remove the acetyl esters. And you can see here that after alkaline treatment, we have also labeling with LM23 in the wild type, and even more labeling in the mutant. So we conclude that there is a difference in the acetylation level of silogalacturonan. We also did an extraction of pectin using pectinases and endopolyglatronase and pectin methylesterase. And what we find was, although there is a bit of variation here, but we find that the 20% decrease in acetate that we see in the starting material is also present in the extract and in the pectin depleted residue. So it looks like there's 20% less acetate in everything we look at. Skip this one and go to this one. So this is an experiment that Sasha in Marcus' lab did for us. So again, here looking specifically at O-acetylation in siloglucan. And Sasha found that in siloglucan from these mutants, there is also about 20% less acetylation. So it looks like, again, every polysaccharide that we have looked at has 20% less acetate in these mutants. I cannot say that this will also be the case for mannans, which we didn't look at, but it looks like everything has the same, and that there's not really a specificity in whatever RWA2 is doing. We were wondering, which we are wondering with all the plants that we are generating, whether they would show any important agronomical changes. I mean, we can look at them and see that they look fine, but what if we take these plants with modified cell walls and put them out in the field? There's always a concern that we make plants that are more easily degraded, so maybe they'll also be degraded when we put them out in the field. But we thought it was important to see if the plants would respond differently to pathogens. We did an experiment with these plants, infecting them with botrytis, which is one necrotropic pathogen that infects Arabidopsis. And actually, we found in this case that the mutant is significantly more resistant to botrytis. Well, that's interesting. We don't know why. We try to see if any pathogen response genes were already induced, but so far we haven't been able to see that that's the case. So we don't know why they're more resistant, but they are more resistant. It's not all pathogens. Candice in Shauna's lab did this experiment where she tried to infect the plants with powdery mildew, and there's no difference in the response. So it's not like they're more resistant to every pathogen. It's specific, and we only looked at those two so far. So RWA2, that's the one where we have seen a phenotype. I don't have very nice expression data to show you, but this is expressed in primary walls or in cells that make a lot of primary walls, whereas the other three, which also form their own little clade here, they have very similar expression pattern, and they're expressed in tissues with a lot of secondary wall formation. And again, we didn't see any change in acetate, probably because they have redundancy. But we have generated double, triple, and quadruple mutants with all these different genes. And the double mutants, they all look normal. The triple mutants, they all show phenotypes. Quadruple mutant is very small and very hard to grow. So the triple mutants, yeah, they look different, and we haven't finished the characterization. But this one, so this is RWA1, 3, and 4, the three genes that are expressed in secondary wall-related tissues. And it actually, I think, to me, looks very much like an irregular xylem phenotype that we have in this plant. And they don't grow very well, but we are, again, in the process of characterizing them. So what do RWA proteins do? We don't know yet. They don't seem to be specific acetyl transferases. That is fairly clear. If you look at the topology, predicted topology of RWAs, they are multi-membrane spanning proteins with 10 to 14 different transmembrane helices. Again, we found them originally as homologs of the Cryptococcus and animal Cas1 proteins. But the homology, the sequence similarity, is only here in the C-terminal part. There is an N-terminal part in the Cryptococcus and animal proteins, which is not present in RWA. There is a weak sequence similarity between this N-terminal part and a group of plant proteins known as DOF231s, which I know some people here in EBI are also working with. This is a big group with maybe 45 or 47 members in Arabidopsis. So we hypothesize that this domain is kind of a transporter domain that transports acetyl-CoA, and it needs to function together with the 2,3,1 group of proteins, which then might be transraces or transesterases. And that the diversity in acetylation that we find in plant cells, where so many different polysaccharides can be acetylated, is mediated by the large group of DOF231 proteins. In humans, we have only one protein, Cas1P, but we also have only one sugar that is known to be O-acetylated, and that is sialic acid. And it's actually recently been shown, there's a paper in Press in Glycobiology that shows that the overexpression of Cas1P, human Cas1P in monkey cells cause an increased O-acetylation of sialic acid. We try to find out what these RWA and also the human proteins do by expressing them in yeast, but I don't have any data on that. One thing that is important for us in JBEI is to kind of assess our technologies to be able to set priorities. So how important is acetate? I told you that it's very important, but how important? Can we put a number on it? And this is important for us to do. So Daniel Klein-Markushammer, and who's working with Harvey Blanche, she's been developing a techno-economical model to try to assess the different things that we work with in JBEI. And the model, and I don't know much about modeling, but I know that they have kind of modeled all the different steps in the production of biofuel. Now, we have to work with something that we know how to do. So his model is based on corn stover, which has a price that we know. It's based on acid pretreatment of the corn stover, enzymatic digestion, and fermentation into ethanol, because these are relatively well-known processes that can be modeled. And then Daniel is trying to put into his model, so what happens if, for example, we use corn stover that would have 20% less acetate, 20% being what we already showed in our Arabinose plant that we could achieve. What would that mean in the end? And according to this model, it will mean that the ethanol price will be 10% less than it was without the 20% reduction in acetate. The reason for the lower price is that actually you can ferment in the presence of acetate, but it takes longer. So you need a bigger fermenter, you need a bigger plant, and you need more stainless steel to make biofuel with stover that has a lot of acetate, and that's the extra cost. Daniel has also modeled lignin reduction. Again, we all knew that lignin was important, but how important? And Daniel tried to put numbers on what would happen if 20% reduction of lignin, and that boils down to a 23% reduction in cost according to this model. So lignin is, of course, very important, and we would really like to make plants that have less lignin. Plants that have less lignin, like these different ones, they have better saccharification. That's great, but they also grow very, very badly. So making plants with less lignin is pretty challenging, and in general, plants with less lignin, they have many growth problems. I would like to mention that my colleague Dominique Loquet has a group in JBEI working with engineering lignin, and they're making a lot of progress with engineering plants that have less lignin and still grow very well. But I will not talk about his data, so you can invite him if you want to hear about that. So we are interested in finding other enzymes. So how do we put the Arabinose on there? How do we put the glucuronic acid on there? And one way that we try to find that was with co-expression analysis. What about the backbone? Well, we know genes involved in making the backbone, although we don't know how it is being made. But there are these three glycosyl transraces, Rx9, 10, and 14, which are all somehow involved in making the backbone. If you knock out either one of them, there's no silane formed. So maybe we could use this knowledge to find other things involved in silane biosynthesis. So we took the three Arabidopsis genes, 9, 14, and 10, and try to look at genes that had the same expression profile in microarray data. So these are published microarray data that we are using. We didn't do any ourselves. And then we did the same with rice, trying to see, okay, the IRX10, 9, and 14 autologs, what genes in rice are are co-expressed with that. And we also try to predict localization, because all the things involved in making silane, they should be in the Golgi. So if we know about co-expression and co-localization, we can find good candidates for other enzymes involved in making silane. And this is just a schematic model that shows then that we find some things that are present in both Arabidopsis and rice in this analysis. So different glycosyl transferase, UDP xylose synthase, and other things. This is the acetyl trend. This is the RWA protein actually in the DOF231 that we find here, both in Arabidopsis and in rice. And then we find some components that we only see in rice and not in Arabidopsis. For example, the RGP protein that I have told you about. Now, Arabidopsis doesn't really have much Arabinose on the silane, so we're not surprised that we don't find that one in Arabidopsis, but only in rice. And then we find things that we only find in Arabidopsis and not in rice. For example, this PGSIP protein that I'll tell you about. So PGSIP means plant glycogenin starch initiation protein. It was published a few years ago that it's located in the chloroplast and it's involved in starch biosynthesis. And it has sequence similarity to glycogenin in animals, which is a primer for glycogen biosynthesis. So it was reported to have this function in plants just related to starch biosynthesis. But we kept seeing this in our analysis, and we couldn't really get it to add up how it would be involved in starch biosynthesis. When we looked at the localization with resin fusion protein and also with Golgi proteomics that we have done and that was done in Paul Dupree's lab, the PGSIP proteins, they are not in the chloroplast, they're in the Golgi. So that already told us that something was wrong. So we thought that this protein is probably making silane somehow, and we looked at the cell walls, and actually the cell walls in mutants that are knocked out in PGSIP, they have a lot less glucuronic acid. And this fit very well with the retaining glycosyl transferase family that PGSIP belongs to. And we could also show that the mutants knocked out in one of the PGSIP isoforms, I have to say that there are 5 or 6 genes in Arabidopsis, that it had a lot less glucuronosyl transferase activity. So what this means is that PGSIP proteins, they put glucuronic acid on the silane backbone. And we shouldn't call them PGSIP, we should call them something else. And this GOX, which I think is glucuronosilane related protein, that has been named by Paul Dupree, who is also working on this family of proteins. And so the final slide here, I will just say that we find a lot of other things, not just things that are directly involved in silane biosynthesis, but this co-expression analysis, we find many things that are in the plasma membrane or associated with the plasma membrane. We find transcription factors. What we are trying to illustrate here is that some of the things that we find in this plasma membrane and related, they are clearly things that are involved in signal transduction. We find similar sets of proteins in mammals. These are proteins that are known to be involved in pathogen signaling. We don't think that what we find in our co-expression analysis involved in pathogen signaling, but perhaps some other signaling from the wall that involves this kind of protein. And we are studying that at the moment, looking at different mutants in Arabidopsis. And this is just a picture of almost all the people in my group. So thank you very much for your attention. Is there another phenotype? Is there a phenotype? Well, as far as we can see, they look normal. Paul Dupree, who has a paper published on this also, he doesn't see a phenotype, but the cell walls are more easily deconstructed. We haven't looked at that, but he has, and it's in his paper. But it's surprising that you can have silane with no glucuronic acid and the plants look totally normal. Don't quite understand that yet.
The RGPs were found because of this effect of reversibly glycosylation. this protein. So how does this fit in with this model, or is it an artifactual side activity that has no physiological relevance when you knock them now all out?
I don't know. Maybe it's part of the reaction mechanism that the nucleotide sugar is transiently bound there and then converted to the furanose form. I can imagine something like that. But then why does it bind all the other nucleotide sugars, which is how it was found in the first place? If it's only working to make mutase activity with UDP Arabinose, why does it bind UDP glucose and UDP xylose, which it does? We have tried to see if there was any mutase activity with other nucleotide sugars. They're not known to exist in the furanose form in plants, but we don't see the activity. Say UDP galactofuranose is known from bacteria, but we don't see the furanose being formed when we incubate with this enzyme. I don't know why. I have a problem fitting this together. It's also a bit surprising that the RGP is actually a relatively abundant protein. Why is there so much of this? All the other proteins that we are working with, the different glycosyl transferase and nucleotide sugar interconverting proteins, they're much less abundant. RGP, there's a lot of it. Maybe it's doing more than one thing. I cannot exclude that.
So I'm not sure what the evidence for IRX 9, 10, and 14 making a linear backbone of Xylan is, but one potential way of getting around the requirement for two transporters would be that the building blocks, with the decorations on them are made outside of the Golgi and then transported in via a transporter that doesn't just transport via the nucleotide sugar or the monosaccharide, but rather the xylose, arabinose, and maybe even something attached to that. So do you feel like that's a possibility, and have sort of multi-sugar transporters been characterized in the Golgi?
Well, I can't exclude it. I think it's an unlikely possibility. We haven't clearly identified the arabinosyl transferases yet, but we have data indicating that they are the GT61 members. And as far as we can see, GT61s are typical glycosyl transferase type 2 membrane proteins with a catalytic site inside the Golgi. So are the IRX 914 and so it doesn't look like we have any of those things outside the Golgi. I think it's unlikely that there would be oligosaccharides formed outside which are transported in. But then again, there are a lot in this mechanism that we don't understand. And nobody has been able to show, and I'm sure a lot of people besides us have tried to show the activity of RX 9, 10, and 14 in vitro, and we have failed, and I'm sure other people have failed too, otherwise we would see the publications. I don't know how it works, yeah.
So, kind of, the model you're saying kind of makes a lot of sense to me. The RGP would be outside the Golgi because you said it has some broad specificity, or so it could mutate, could interconvert multiple sugars, right? No, it can only do
the RGP?
Yeah.
Well,
you showed HPLC tracing.
The RGP converts UDP arabinose between the furanose and pyranose form, and we have not been able to detect that kind of activity with any other sugar. But it's known to bind nucleotide sugar. So if you incubate RGP with radioactive nucleotide sugars, not any, but any that people have tried, they bind them. And you can run it on a gel and you have a radioactive protein. Then if you incubate again with non-radioactive nucleotide sugar, you can chase off the label. So that's why they're reversibly glycosylated protein. Why they do that, we don't know, because it doesn't seem to fit into the reaction mechanism of converting the pyranose to the furanose form. So I cannot understand how these things make sense together.
Is there any way to localize the RGP to the Golgi?
Well, the RGPs consistent with what we see with our fluorescent protein, but also what has been shown previously with immunogold labeling. They are cytoplasmic proteins on the outside of the, or associating with the outside of the Golgi. Not very tightly. You can wash them off, but you can find them on the outside of the Golgi.
But there's no like localization signal or something, so you can actually get it inside of this is to see if perhaps it would be detrimental to have it in there where all the other sugars are, I guess is what I'm saying. Although
well, we could, yeah, I mean, we could try to put it inside the Golgi and see that would happen. Yeah, that might be an interesting experiment. Well, sure, you could just make a fusion protein with the transmembrane region from another glycosyl transferase. That experiment, I'm sure, could be done. I don't know what would happen. It might be worth giving it a try.
What organism was in the techno-economic model? What was doing fermentation?
The techno-economical model is developed with corn stover. You asked organism, didn't you?
Yeah, very end. What was making the ethanol?
Oh, yeast.
I was just wondering, when you were discussing those nucleotide binding studies, were those UDP and GDP-linked sugars, or was it the only UDP nucleotide sugars that it was able to interact with? Maybe it's binding UDP as opposed to...
With the RGP protein? I mean, we only checked UDP sugars. I don't know if anybody has actually seen if there's any reversible glycosylation with GDP sugars. I would expect there wouldn't be, but I don't know if it's been tested.
Maybe one more question for Brad. The DUF231s and Cas1P homology, is that predicted to be inside or outside the Golgi?
Well, in my prediction, the DUF231s are also type 2 membrane proteins with the catalytic, well, or the main part of the protein inside the
I'm wondering
Golgi.
about the Cas1P because that's transmembrane domain.
Yeah, I get this. I get the same prediction there, but you know, it's a little bit difficult to say exactly how many transmembrane helices there actually are in Cas1P, so I don't think we know for sure.
Is that portion the proposed catalytic site for DUF231s? Or is that some other portion of DUF231s?
Well, the DUF231s have a site that looks somewhat like a esterase, right?
Is that the part that shows homology?
And that would probably be inside the Golgi, but I think I mean, the evidence for that is not really, I don't think we know for sure. But in our fusion proteins, it is inside the Golgi, right?
Right, same with mine.
Yeah.
No, I'm just wondering, the homologous part of Cas1P, is that the predicted catalytic site for between DUF231 and Cas1P, or that's spanning the predicted catalytic site for DUF231?
Yes,
Okay.
yeah.
All right. Thanks. Do we have any